Hello, I'm Ryan, an applications informatician with Oxford Nanopore Technologies. As a member of the applications group, I research, develop, and support software tools to enable experimental application scientists. In this video, I'll be discussing one of the many exciting applications made possible with Nanopore technology. The applications group explores and demos uses that were previously difficult, if not impossible, with existing short-read, fixed runtime technologies. We exploit the natural advantages of nanopore sequencing on the MinIon platform. It's portable, it's single molecule, it's long read, and it's real time. For more information about apps, stop by our posters or have a chat with one of the members of the applications group. In this talk, we'll be discussing a specific type of sequence difference called a single-nucleotide polymorphism, a SNP for short. Let's imagine two alleles, lovingly termed 1 and 2. A single nucleotide polymorphism is a single base substitution, for example, a mutation from an A in allele 1 to a T in allele 2. While a single base change might not seem terribly important in the context of a megabase or gigabase genome, SNPs are important. First and foremost, SNPs are biologically relevant, being implicated in human diseases such as beta thalassemia and cystic fibrosis. Additionally, SNPs have also found use in evolutionary genetic studies and human identification. Furthermore, there are exciting emerging uses for SNP detection. These include cancer diagnostic tools and the evergreen promise of self-quantification and personalized medicine. Expanding on the musings of our CTO, Clive Brown, somatic SNP detection in your toothbrush maybe. To learn more, check out Clive's recent talk at Wired Health. For a specific example of human disease, consider sickle cell anemia. A single base mutation in the gene encoding for hemoglobin, an A to a T, causes abnormal development of blood cells, wherein the usually flexible cells become rigid and sickle-shaped. These rigid cells may block capillaries, leading to the familiar clinical symptoms of severe pain and anemia. Cancer typing is an example of the emerging diagnostic use of SNPs. For example, many SNP variants of the tumor protein 53 gene are implicated in cancer severity and progression. Curiously, most p53 SNPs are located outside of the coding regions of the gene. To detect SNPs, most common bioinformatics tools rely on consensus. For example, aligning short-read next-generation sequencing data to a known reference, and then reporting the consensus probability of each base variant. Base-based consensus approaches are of course possible with nanopore data. However, tools designed for short-read data are not necessarily optimized for working with long-read, real-time nanopore data. Metricore, a real-time cloud computing service, is engineered for nanopore data. Indeed, consensus SNP detection is available via the Metricore platform. For more information, please visit metricore.com or attend Breakout 3, Nanopore Field Trip, to hear more about how nanopore technology, including consensus SNP detection, has been applied in the field. Nanopore sequencing detects changes in current related to the bases occupying the pore. By detecting events in the current signal, we reduce our data to a squiggle space representation. This reduced squiggle space data is then base called. Using a model, which contains information such as the characteristic current level for each set of bases, we translate the squiggle space event series into a base call. This is accomplished using a hidden Markov model of all possible transition paths. The base call is the maximum likelihood path. Alternatively, we can constrain the transition path and evaluate which of multiple fixed paths are the most likely given the data. For more information about constructing your own models, please see the applications Pechakucha entitled Custom HMMs, designed for your biological context. The change in current is caused by bases being passed through a constriction in the pore. Due to the finite size of the bases and the pore constriction, the current level of each squiggle space event does not correspond to a single base, but rather a set of bases. This set of bases is called a k-mer. Because the DNA translocates one base at a time, a SNP will be detected as present in several consecutive k-mers. This is why the squiggle is affected over several consecutive sequence positions. For example, we show the squiggle space view of 4 possible SNPs for a particular sequence. We use this additional information to more accurately detect SNPs with nanopore data. It is important to note that characteristic current levels differ for each k-mer. A SNP from an A to a T may look very different depending on the surrounding sequence context. With squiggle space data and a model of the k-mers corresponding to each base, we can use mapping to classify which transition path is the most likely. Importantly, this yields a single molecule estimate for each individual read. In this case, C is the most likely base. By aggregating single molecule SNP calls from many reads, we can also generate a consensus. For example, we plot the single molecule SNP estimate for 100 single molecule reads. Each read is represented by 4 points, probability estimates for A, C, G, and T. Unsurprisingly, the consensus prediction is also C. Importantly, the individual read estimates contain negligible false positives. For a sample of this size, at greater than 95% confidence, there is no false positives for A, G, and T. In other words, if a SNP is detected with high confidence from a single molecule, one can be fairly confident in the result. Moreover, the majority of individual reads may be classified to greater than 95% confidence, indicating that throughput is not sacrificed to achieve a low false positive rate. With the MinIon and nanopore sequencing, robust detection of single base changes in single molecule reads is a reality. Imagine applications such as real-time strain identification, detecting natural base modifications, or wholly artificial unnatural nucleotides. The possibilities are endless. The Applications Group thanks you for listening.